Okay, the final sprint of the day. So we have a couple of more sessions coming and then some brainstorming with Paul later on. So this session coming up is on mammalian biofoundries. So once again, this is a product or result of putting me in charge. So I thought this was really important. And I think we saw bits and pieces throughout the yesterday and today. We saw something flash quickly from Mark on the Imperial Foundry. We saw something flash yesterday quickly on the Edinburgh Foundry, setting up mammalian capacity. I know there's people here from Genopole and France that are really interested in developing that capacity. So even though it's not a major activity within the GBA, it's definitely is a growing opportunity. And there's some early actors in the space that are trying to forge new grounds. So I thought it would be important to expose this emerging space to everyone in the GBA. We at Concordia spent quite a bit of time in the last, I don't want to say how long, but for a while trying to set up a platform, automation platform to do this, only realizing when you went outside and started speaking to other entities, this is a really rare and difficult thing to do. So I think there's a lot of cool technology and lessons learned potentially that you're going to get from this. A lot of you have been to the Concordia Foundry, so you've probably seen some of the systems that we have there. But hopefully you'll have time to ask more questions and get more answers. So we have two different speakers today, and hopefully they complement each other. So I'll introduce the first speaker and then the second speaker after that. And hopefully we'll have a little bit of time for questions afterwards. So I want to introduce our first speaker for the session, which is, we'll call him Kevin number one, because it turns out we have two Kevins in the session. So Kevin is a research associate in the Foundry. Joined the group, I want to say about a year ago, January this year, I think, is what it was. Yeah, so it hasn't been that long, but he's been very instrumental in leapfrogging and pushing our platform forward to the point where it was operational until the Vantage blew up last week, but operational and functional. And he's going to show you what that looks like today and hopefully talk about how we move forward from there. The floor is yours.
Yeah, thank you. Thank you, Vincent. Hi, everyone. So like Vincent introduced, I'm Kevin, platform coordinator at the Genome Foundry on the mammalian side. And I'm going to give you today a quick overview of what we are doing on this side of the Foundry, if you didn't have a chance yet to get a tour of it. As you know, the biology is becoming more and more complex. Only a small fraction, for instance, of the human genome is currently being studied. Some will say around 10%. It's true also for the subsequent proteins that comes from this genome. So there is really an urgent need to combine biology with automation to accelerate the development of new methods and accelerate discoveries. And this is why biofoundries were created, and this is why we have biofoundry here in Canada. Our biofoundry is very unique because we have two sides. The microbial side, where we mostly do DNA assembly, sequencing, and also microbial technology, along with FACS, everything in high throughput. And we also have the mammalian side, where we do mostly iPS editing and advanced tissue culture. And behind this, this entity, there is an amazing team with Nick and James being part of the microbial side, Angela and myself managing the mammalian side, with Vincent and Audrey, that I would like to thank today for organizing this event, and Ben also, that you might have speak with, business advisor. So that's very amazing team that's managing this unique platform. But today I will focus more on the mammalian side, on which we have one big robot, one big guy, which is called the Hamilton Vantage. And what's very unique with this system, it's like you can see the robot over here, and it's inside its own enclosure, which is a biosafety cabinet. And the whole system with the enclosure and the robot inside, it's connected to a Lyconic incubator. and controlled using a particular software that I will showcase in a few slides. And what we did, it's also, I will say, kind of pimped the deck to make the workflows run smoothly. We also installed 3 cameras, 2 on each side, to monitor what's going on the deck, but also a camera on the deck, because the Vantage has different levels, as I will explain in the following slides. And a very interesting feature, it's like we installed a software to be able to remotely control the Vantage from anywhere in the world. And as a joke, for instance, I was able to control it from my swimming pool and do experiments from my swimming pool. And we'd like to perform the first transatlantic experiment with some coworkers in Japan, where they will be controlling the robot from Kyoto, and we'll be here ready with Angela, managing that everything is going well. So to give you a more in-depth overview of this robot, we have 2 decks on this robot. The upper deck that it's on the top over here, containing multiple elements, and the lower deck, which comes empty when you buy the robot. The upper deck is basically like a bench on which you can perform experiments. You can see over here the tips, a tilt module, because when you are at your bench and you want to remove media, you need to tilt the plate to be able to properly be able to remove all the media. We have some reservoirs on which you can put any type of media that you want, enzyme. We have a heater shaker. And the 2 main units of these robots are first, what we call the ML Star module. Basically, it's a multi-channel pipettes, which contain 8 channels that can pipette between 1 microliter and 1 ml. And we have 2 5 ml channels that can pipette between 1 ml and I will say 4.7 ml. So this unit is moving all along the deck and perform all the pipetting. We also have the track gripper, which is kind of like a claw that is able to go around the deck at 360 degrees and grab different elements, plates, and move them all over the deck. As you can see over here, the track gripper is performing action like placing this plate, removing the lid, and put it on this plate. And ML Star is doing all the pipetting over here using 1 ml tips in this case. We have the tilt module over here and the heater shaker, which is located on this part of the deck. And that summarizes what's mostly present on the top of the deck, like mostly like a bench. And the very interesting part is also what's underneath, what we decided to put. In our case, we put a Citation 5, which is kind of like a micro imaging station that can take automatically pictures of cells with bright field, phase contrast, BFP, GFP, and Texas red. And we also have space for another wavelength by inserting a cube that we'll have to buy eventually. We have a cap decap module to freeze the cells. This unit can basically load a box of tubes on which cells were put, cap the tubes. Each tube has its own barcode, and once the box is capped, can be dispensed and put back on the deck, and the user simply has to take the box and put it into the liquid nitrogen or at -80. So no need anymore to do tube by tube, which can be pretty long. We also have a Lanza 384 unit to perform nucleofection of the cells. So far, nucleofection is one of the best way to deliver DNA and even native protein to the cell. And we also have an entry-exit module, which basically is a drawer on which you can put a stack of plates, and the entry-exit module will read and count how many plates were inputted inside, and can even read in some cases barcodes that we put on the cells. So on the plate, so no need anymore to label each plate. So we designed a unique system which virtually allows like high throughput tissue culture and many experiments. That's for the part, really the hardware part. there is the part which is mostly the software, which is extremely complicated in this case, because we're using a software which is called VOV, Venice Advantage. And this software is building a method with this software is a multi-layered process. First, we need to build the virtual space which correspond to the environment on which we're going to work. Then we need to create an in silico version of our deck, because the deck is very flexible. You can put the tip on the left, the tips on the right, etc. So we need really to model the deck accordingly to what we physically have in the lab. Then we need to put different type of labwares that we're going to use. Will it be 6-well plate, 24-well plate, etc.? So tubes. Then comes the very complicated part that we need really to tell the robot what steps to perform. The robot will do exactly what you tell him to do, but it needs to be really well defined. Like take the plate A, move it position B, shake it, pipette, etc., with condition. And since it's automation, we also need to deal with many issues and many things that can go wrong that I called over here like Murphy's Law. When something can go wrong, it will go wrong. And the best example that we had with my coworker Angelites, we simply asked the robot to remove the lid of a plate, but the lid was too tight. So the track reaper came and took the whole plate at once. So for those who had the tour of the foundry and had the chance to take this plate, it's fairly heavy. So this plate can really fall at any time on the deck and break any parts of the robot, which are fairly expensive. And we have plenty of examples of tiny details like that, tiny things that can go wrong. And we started a list of things that we became pretty much expert on troubleshooting those issues. Here is an overview of how we create a method. So on the left side, you have the virtual space that we're going to build, and for the robot to identify on which environment is going to work. And on the left side over here, you have all the commands that basically it's just drag and drop, but each step needs to be identified. So you can see over here that I'm building a deck and putting all the elements, the tips, the garbage, liquid, some elements, and then I'm starting to build the method by dragging and dropping stuff. And windows are very complex with advanced parameters. So it took us some time to really get to this part. And once we are done, it's around 2,000 lines of code that we can spread into multiple tables. So that's the power of the software. It's very powerful when you know how to use it, but the con is like it's very technical. It's very technical and challenging. So the way that we decided to tackle this issue and to get very familiar with it, it's to create micro methods. Because as I mentioned on the deck, for instance, you have a pipetting unit, you have a heater shaker module. So we started with the pipetting unit and say, okay, can we move it 1 millimeter on the left? Okay, it works. Can we move it 30 centimeters on the left? Okay, we can move it safely. We know how to do that. Now, can we move it and pipette? So on and so forth. So we develop kind of like blocks of code that works. And once we got local control over all those elements on the deck, it was just a matter of arranging them to create a working protocol. And those parts, we're trying to make them as flexible as we can. Since, for instance, when you want to passage cells, 293 versus iPSCs will be fairly the same protocol. What can change is the time that you leave your enzyme on the cell. Do you want to shake the cells? Do you want to rinse the cells with PBS before putting the enzyme? So we are including all those options for the users to be able to have a single protocol, and for us to do not reinvent the wheel every time that a new user is coming. And you can see over here one of our protocol, which is very complex, where we remove the lid of a plate, we remove the media, we change the media, and over here we put some DNA, and we're gonna prepare the DNA to perform a nucleofiction in this Lonza plate, and the plate will be taken down under the deck to perform the nucleofiction. So we believe that understanding what we are doing and having a very robust environment and a lot of knowledge about it will allow us to onboard new projects. And once we were comfortable enough with this robot, we could perform our first semi-large scale assay in collaboration with Dr. Wolstein from the University of Kyoto, which was producing and testing an array of 96 PEG RNAs. So as you know, prime editing is fairly new. It was really released 3 years ago, and it relies on the unique Cas9 PEG RNA system that you can see over here. And what's important is the composition of the PEG, how the PEG is designed. And the PEG contains 2 different regions, which are called the PBS for the prime binding sequence and the RT for the reverse transcription. Despite people using this system, at the time, little was known about how those regions, PBS and RTT, can affect the prime editing efficiency. And there is a simple assay to evaluate this efficiency, which is called GFP to BFP conversion. Simply put, we can convert the GFP into BFP using a single mutation that will change a tyrosine into an histidine. So one could basically create a cell line which express GFP and design a PEG meant to do this specific mutation. If the PEG is efficient, then the GFP will be converted into RFP. If the PEG is not efficient, then the population of cells which are GFP will remain GFP. And this efficiency can be assessed by FACS, where we plot GFP against BFP. If there is no conversion, then all the cells will remain GFP. If there is conversion and the PEG is working, then we'll see a population of GFP cells. And this was the idea behind this assay, where 96 PEGs were designed by the Dr. Wolstein's team, and the PEGs were produced on the microbiome side. And we got a plate of 96 PEGs where each row has a PEG having a given RTT length, starting at 7, but having different PBS length from 8 to 22. And each column contains a PEG having a different RTT length. We performed this experiment in triplicates, meaning that each PEG was at least tested 3 times, and we made more than 250 cell lines in a bit less than 6 weeks. And the only thing that we knew when we got this library of PEG, it's like the PEG E6 was a positive control, a PEG meaning that's working and converting efficiently the GFP into BFP. So before presenting you the data that are on this graph, I will just take a few seconds to quickly explain the way that we put the data. So on the top, you will have the groups. For instance, the group on the lane on the row 1, A1, A2, A3, A4 till A12. So this is the group A on the first row, having RTT of 7 and different PBS. And then we repeated the graph in this way to have different group B, C, D with different RTT length. And for each RTT, a different PBS length. And this is the kind of results that we got. Conversion going from between 0 and up to around 40%. And what it's very interesting, the first thing that we can see, it's the PBS does not seem to impact the prime editing efficiency, because each group A over here is really not efficient at converting the GFP into BFP. Conversely, we can see that the group E is the most efficient. And this was performed like recently, and in 2021, there was a big paper published in Nature Biotech where they did the same thing with 4,000 PEGs that was pooled. And they came to the same conclusion where the RTT is really important and more important than the PBS length, and the best RTT is around 12, 13 nucleotides. So exactly what we found. And we also pick some candidate, cherry pick some candidates from each group, and we perform like fluorescence imaging. And as you can see over here, we have a negative control with the cells endogenously expressing GFP. So we have no BFP. PEG A9, which is one of the worst performing PEG over here, we don't see any GFP. But when we took one of the best PEG, E9, we could see indeed some GFP cells and around 30–40% of the cells. Even though the contrast intensity is not that good over here, every time that you can see an arrow means that a cell was successfully converted. And finally, we confirmed that the PEG E6, which was the positive PEG used by the team in Kyoto, is working in our assay. So we basically, with this assay, hit two birds with one stone. It allowed us to validate our system using what was published in the literature and what was already known from the Kyoto's team, but also test the whole library, what they couldn't do in Kyoto because it was too much work. But basically, establishing a working protocol is just the tip of the iceberg, because this is performing an experiment on deck. And what we want to do also is be able to do automated imaging. You can see over here, as I mentioned at the beginning of my talk, we put 3 cameras on the top of the deck, 2 on the top of the deck, sorry, and 1 at the under deck. And you will see, for instance, over here, the citation 5, the tray going out and the track gripper taking the 6-well plate and putting it inside the citation 5 and taking picture with the user only clicking on one button. Sorry, I'm gonna start it here. So you can see the tray going out, the track gripper taking the plate over here, putting it on the citation 5. The citation 5 is taking the pictures, each well, and bringing back the plate on the deck. And for the citation 5, we have a software where we can create protocols. For instance, take one picture on the middle of the well, only phase contrast or bright field, or use the GFP and take 4 fields, and you can basically create any type of imaging protocol. And then the user can come back the next day and we'll have like a template of its plate with the different condition and different wells, and can just take a look at the cells. We also are working on automated imaging and automated cell culture because the citation 5 can also evaluate the confluency and automatically count the cells, live cells. So virtually we could take a plate from the incubator, put it in the citation 5, and create a workflow that if the cells are too confluent, then passage them. If not, just change the media. And this is what we are trying to create actually. A file that will allow the robot, the Vantage, to communicate with the citation 5 and communicate also with the incubator. Like I mentioned, take a plate from the incubator. If confluency above 70%, execute cell passage method. Otherwise, change the media. And we could create a nested process of if or else functions to manage those cells. And all of that thanks to our iconic incubator that you can see over here, which contains a carousel that can turn over here. And we have a trap between the incubator and the robot over here. So where the robot can put a plate, and there is something like kind of a pizza tray, very thin, taking the plate and getting it inside over here on this way, with a system that takes the plate and switch it and put it on the proper location. So that would be one of our challenge, to create this interface between those 3 robots for them to communicate and for us to keep track of all those cell lines. And another piece of the puzzle that we are working on is to create a proper user interface, because at the Genome Foundry, we are also here to help students and help them to perform their experiments. And we do not expect anyone to come and start to code their own stuff. So we are doing it for them. And for instance, we can take a huge block of code to create a simple panel, which will be kind of like a sandbox where the user can come and click on the simple button and the protocol will be executed. For instance, we could create a button which is passaging the cells. Then a window will pop up. Would you like to rinse your cells with PBS? Yes, no. What is the volume of enzyme that you would like to put? how long would you like to put the enzyme? Very standard questions that any student familiar with tissue culture will do without even having a look on the code behind and knowing how it works. Do you want to take a picture of your cell before, after, both or no, etc. And very importantly, get the feedback from the students to know if something is missing on that. And I would like to give you a few numbers before finishing this talk. Currently, we can perform around 50 edits a day. By the end of the year, beginning of 2023, we would like to be able to perform more than 400 edits a day. Because currently it takes us around 40 minutes to do 24 nucleofiction, we identify the bottlenecks along the pipeline, and that will be something that we'll be working on over the next months. That could lead us to, I would estimate, process a 24 well plate with the current setup that we have at around 15 minutes. And there is still a way to upgrade our system to even go faster. And we are currently working with one type of cell lines, which are iPSCs, because they are very picky and hard to work with. So once we'll be set up with this cell line, technically we can onboard any type of cell line. It shouldn't be a challenge to get 293 or U2S cell lines on the deck. We currently have 2 working methods and only 2 users, but thanks to this master panel, we start getting a bit more users. And currently we can manage around 50 cell lines at a time, but we could virtually go with thousands and thousands of cell lines. And if we fully fill up the incubator, it would be 30,000 cell lines. Something that I don't think we'll reach at some point, but this is the capacity that we can reach. So I hope I convinced you that we are currently building a platform with which is using cutting-edge technology, and that will allow high throughput mammalian cell culture and many advanced techniques. And we are currently developing this workflow for iPSCs, but as I mentioned, it's really it will be ready made for any type of cells. And this pipeline could be virtually applied to any types of cells. And as I mentioned, editing, imaging, and even more. So with the goal of engineering the biology at the next scale. And on that, I would like to thank you for your attention, and looking forward for the brainstorming session. Any questions?
I'm debating whether or not we should hold questions for the 2 talks, or is there questions now for Kevin number 1? Oh, there's a quick question coming.
Gonna try to not break this thing.
So thanks for a nice talk, Kevin. So if you get up to that point where you're handling 30,000 cell lines and taking pictures all the time, you're going to end up with a very, very large data set. Have you thought about the fact that that might be a resource in and of itself, and tracking good metadata and stuff like that, so that you can then potentially do some kind of deep learning on these images?
Yeah. We currently are developing with our bioinformatician, Flavia, part of the team, a LIMS system wherein we can keep track of all the samples. It's mostly used right now for the microbiome side, but this is something that we'll implement on the mammalian side. We'll stop working with label plate, but everything will be barcoded. And if you did a tour of the foundry, you saw that we have barcodes below the tubes that we are freezing. We have barcodes on the side of the plate. And the good thing is, like, we won't go from 5 cell lines to 30,000. So this is something that will gradually improve. And at some point, we'll probably hit some bottlenecks. And then we'll have probably to hire more people to find new technologies to process all the data that will come from that. So this is the way that we worked so far, step by step. We made it work with 1 cell line, then 20, then 50. We are scaling up. But yeah, this is something that we'll have to solve on that we are already thinking of and solving already.
But yes, we are thinking about it. I guess your underlying question is, are you thinking about some AI ML kind of approach to image analysis on that scale?
Yeah, cell, like the cell line, morphology, density over time. I mean, you're going to really be gathering a lot of data.
Yeah. So we haven't started that yet. but it's definitely in the back of our mind. And solving the data, these files and all of these files, it gets pretty big. So this is something we're also addressing in terms of data storage.
This is something that we can do with the citation file. We can do phenotyping. For instance, we could see that IPS are not IPS anymore because they are differentiating back into fibroblasts. So we can apply this filter, for instance, when we check the plate. And if like more than 5% of the cells are fibroblastic, then on this file that I described, this interface between the three robots, we could get a red formatting condition. Oh, this plate or this well is not good anymore. And yeah, the idea will be to have this Excel table that will be managed automatically and become red or green over the time. Yeah.
All right, we'll have opportunities, I think, for more questions afterwards, but please join me in thanking Kevin for a great talk.